So thank you, Patrick, for this nice introduction on the field, and thank you for inviting me together with Beat Loeffler for this wonderful conference, as usual, every year. So yeah, it's a great pleasure as a coordinator of NanoAthero project to present the work which is done by this consortium. And I will go fast at first slide because Patrick already presented the field. You are all aware that it's a major issue for health because it's still the leading cause of death in the world. So it's a complex disease, and as it was presented, you have many cell and biochemical processes that are occurring during the evolution, during the age, and they are still a very important issue to tackle when you want to prevent, to detect, or even to treat this major event like plaque formation, plaque rupture, or thrombus formation. And it was really the goal of the project, and it's what we are working on. So we managed to have a large consortium in Europe, 16 partners from 10 different countries. We started 3 years ago, and what was interesting is that we tried to gather complementary expertise ranging from academia, industry, and clinical center to have all the expertise that we need to have the development of a nano project, nano construct for atherosclerosis disease, and I will tell you how we have organized this process. So in fact, we went quite nicely, starting from the design of a nanosystem, their preclinical evaluation, including in vitro toxicological studies, GMP production of the nanosystem, regulatory and toxicological files, and clinical trial. Together, of course, we have risk assessment and ethic consideration all along the project, together with dissemination, training, and exploitation. Of course, because it's a large project, we have also a lot of work for managing of this type of project. But what is important, this is a regular flow, but in fact, we have started to work both on these two work packages, but we have already, at the beginning of the process, started with nanosystems that were already at the beginning of GMP production. Also, at the beginning of the project, we had also one nanosystem which was ready to enter the clinical trial. So I will show you already the results that we have. First, with the design and validation, preclinical validation of the nanosystems. So this process are still going on. Then a few steps on GMP production, and already clinical trials that were obtained with nanosystem in the cardiovascular field. So, as you all know, a nanosystem could be very interesting, especially in cardiovascular disease, when you want to see what is going on for plaque biochemical event, biochemical activation, cell activation, and also thrombus. So it's why we design several types of nanoparticles, and both with the idea to have the diagnostic for the imaging agent or drug inside or outside this nanosystem. Of course, you have to prepare the right design, so you have to think about the structure of the nano construct, and also in some cases the targeting potential of this nanosystem to have a specific activity, and I will show you in one example how it could be interesting to have this opportunity. So in fact, we started, it's a large project, we started with several types of nanosystems, and most of them are represented here. And several partners have already a good experience coming from very, very small nanosystems that are macromolecular systems until micro systems. So ranging from iron oxide nanoparticle, micelle, HDL, lipidodes, liposome, and so on. So we all combine this, but instead of testing only one for one application, we set up a large set of evaluation for candidate nanosystem for 4 indications. I will come back to the 4 indications that we are dealing with. So our purpose, it was to have a table of the parameters that we need, and we selected 7 main parameters. So ranging from here to here. So this is mostly for the design of a nanosystem, going from the physical, chemical characterization until stability, evaluation, preclinical evaluation, for going to pharmacokinetic property, purity, and so on. So for all the system and for the 4 main applications, we try to have this table with the report of all the data we need to select the right candidate for the nanosystem. So again, instead of having only one system for one application that could fail, we have the table for all the nanosystem and the data that we want to have to check, to select or not the candidate nanosystem for the right application. So it's what we are doing. So many, I will not go in detail, but we have several nanosystems, and we have even more. So we have several types of targeting agents, several types of compounds. So we are doing this. The 4 lines of application are thrombus, both for imaging and treatment, and plaque, both for imaging of treatment. And for all 4, we were starting with design on preclinical validation, GMP, on toxicological data, CARPA assessment, and so on. And we have this, and until clinical trial in some of the indications, and I will show you the first applications that we have. So let's start from the beginning. Thrombus, imaging of treatment. So we wanted to have an impact and to have here a clinical trial for the imaging of thrombus. Not yet for diagnosis, for the treatment, it's what knows the purpose until going to a clinical trial, but we are working on it. So let's start from imaging of thrombus. The idea is to design a nanosystem which has a targeting capability to have something to detect the thrombus. On thrombus, it's a platelet activation. On platelet activation, you have P-selectin activation. So we were having one system, which is fucoidan, which is a natural polysaccharide that has affinity, very high affinity, nanomolar range for P-selectin. And P-selectin is expressed in activated platelet, not in a resting platelet, but only in activated platelet. And with this, we developed several types of nano and micro system with the targeting of fucoidan. So in this spec imaging image 2 hours in OAT, we were able to really detect the thrombus here. When you have fucoidan, when you have no fucoidan, you are not able to see something. If you have fucoidan but you have a healthy rat, so no thrombus, you have no detection. So you have something specific. And this is a microparticle made of polysaccharide without made in aqueous solution, absolutely biocompatible, labeled with technetium and coated with fucoidan. We have developed several types of nano and micro systems with the same concept. Here is an example of iron oxide particle coated with dextran and with a targeting moiety with fucoidan, and in vivo, again with the same model, thrombus in the abdominal aorta. We were able to clearly detect the thrombus when we have fucoidan. We have, of course, many controls showing that only the fucoidan can target the thrombus. And if you load this iron oxide particle into a microparticle, but then you cover the surface of microparticle with fucoidan, again, by MRI, you can detect the thrombus in the rat. So these 2 data were already published. So now we have demonstrated that we have a targeting and we can image the thrombus. So we are working on this, and I will show you what we are doing. But because we are targeting the thrombus, we are starting to go for the treatment, and because we can target the thrombus, can we load something? And it's what we started to do, especially with in vitro experiment, especially under flow conditions. So we designed this type of assay. So using a micro system and nano system again coated with fucoidan for the targeting, on having a load on the surface with TPA. TPA is a clinical product which is used to solubilize the thrombus. So instead of having the free drug, we coated the surface with both fucoidan and the TPA, and we were able to see in vitro first by this flow chamber experiment under high or low shear stress that fucoidan loaded with microparticle and with fucoidan at the surface are able to detect activated platelet as compared to non-coated nanoparticle. And this was observed also in vivo in this thrombus model in the mesenteric vein in rat. Here you can see the platelets. They are labeled in fluorescent in green, and the microparticles are loaded with a fluorescent red dye. So we can colocalize the microparticles where they are coated with fucoidan, but not when there is no coating. And these results were very efficient because the drug and the nanoparticle is present on the thrombus because of the targeting. And then the TPA is active and much more active when it's on nanoparticle as to compare to the free drug. And this is quite significant. And we have really promising results even with a low dose as compared to the free drug. So here we had 4 times less than the free drug and much better efficiency. So this is in vitro data that are really promising. So we are going to push this type of product even furthermore. So to make a short summary only on this part, so now we are working on the imaging of treatment of thrombus using a targeting moiety. And this targeting moiety allows to decorate a nanosystem, several types of nanosystem to detect the thrombus, and also to have a loading of a drug. We are clinical already approved a drug for tackling the thrombus and to try to solubilize the thrombus with a much efficiency than the free drug. So we expect to continue and to work on this until the clinical trial. Let's go a few data on plaque treatment. As I said, we have 4 lines of application. One is plaque treatment, so also with a partner in Twente, we have developed, they have developed a liposome construct with several types of drugs. I cannot tell you more about the drugs that are under explanation, but under investigation, but they are working in progress to see the effect of this construct. They are working, of course, of the formulation of this nanosystem with all types of drugs that are listed here about the stability, the construct, the efficiency, the polydispersity, and so on. All these physical parameters, also stability at 4 degrees, at 37 degrees. And each time we try to select the best combination, the drug and the platform, which is interesting. So this work is in progress. And because we have now already some quite interesting preclinical data, we are moving for 2 types of product under GMP production. So this started this year, GMP production of the macromolecular systems that you have seen labeled with technetium for thrombus imaging. And now we are able to produce this compound under GMP condition, and now we will enter regulatory toxicological assay together with a partner involved in the program, and we are hoping to launch a clinical trial at the end of next year. Again, a liposome, as I said at the beginning, a liposome preparation with a drug loading was already available at the beginning of the project together with partner 8, 5, and 14. So they were able to have a pharmaceutical GMP grade of nanotherapy, which was published, and then with a partner AMC, which is Eric, the last presenter of this session, they were able to see if you have the delivery of the liposome to the plaque, and it's what they have seen. So here it's a pharmaceutical assay of this loaded liposome in humans. So 7 patients were injected with liposome as compared to 7 as a placebo, and they were able to see the pharmacokinetics of this nanosystem in human, and then to have a correlation of delivery of the liposome in the plaque with the macrophage content into the plaque. So this was a proof of the delivery of a nanosystem in human, which was not so often. So this liposome accumulate in human macrophage under in vivo after intravenous administration. So this more work is going on. Some of the work I cannot tell you, but we have many working systems under consideration, so I cannot tell you more about this. But it's a very interesting proposal, because we mix all the expertise we need from the design of particles until the clinical trial, and taking into consideration, of course, all the ethic, regulatory consideration, and to tackle cardiovascular disease. So, of course, we are also working on training, especially training on youngster scientists. We had the first session at the beginning of CLINAM. We have many training sessions for days, weeks, or months on different sites. So scientists can work in several areas, in research, preclinical center, in industry, inside the NanoAthero project. So it's very helpful for them. Of course, we have dissemination. We have already more than 20 internal publication for this. We have also publication with Panos. Panos will not present the talk later on, so I have still some time left, and we have also both working on these 2 projects on atherosclerosis disease and management of them with a nanosystem. So this is my conclusion. I still believe that really nanomedicine is a great opportunity for both the diagnosis and the therapy of atherosclerosis disease. It's a challenging issue. We have to deal with chemistry, biology, bioengineering, and pharmacy. And of course, we need a lot of expertise going from risk analysis, ethics, toxicology, and so on. It's what we are working on. So it's like a big project, a big lab, all working together to have this development. And I would like to thank you. And we are also making art from this nano system. Thank you. I think it's a question at the end. Okay.